Hello children, this session is a continuation of our lesson Organization of Tissues. Now in this session we are going to see the cell division. Cell division. You may wonder how a single cell then goes to form such a large organisms. All cells reproduce by cell division and division of cells into daughter cells. So this is called the cell division. Now let us move on to the types of cell division. So the three types of cell division that occurs in the animal cell are amitosis, mitosis and meiosis. So what is amitosis? It's a direct cell division. Mitosis is indirect cell division. Meiosis is a reduction division. Now let us see what is amitosis. Amitosis is the simplest mode of cell division. It is the simplest mode of cell division. It commonly occurs in unicellular organism. It occurs in unicellular organism. During this amitosis, the nucleus elongates first. So in the first picture you can see the nucleus gets elongated and then constriction appears in it, which deepens and divides the nucleus into two. So what happens here? First the nucleus elongates and the constriction appears along the sides and the nucleus divides into two daughter nucleus. Followed by this is the cytoplasm divides, resulting in the formation of two daughter cells. So this is amitosis. I am repeating. So in the first picture you can see the nucleus gets elongated and in the second picture a constriction on the wall of the cell occurs which enables the cell to divide. So first division occurs by the nucleus followed by the cytoplasm, the division of cytoplasm. Here amitosis is one cell divides into two daughter cells. Are you clear? So this is amitosis. Are you clear children? Next we will move on to the mitosis. Mitosis was first discovered by Fleming in the year 1879. So in this cell division, one parent cell divides into two identical daughter cells, each with a nucleus having the same amount of DNA. Are you clear? So here the mitosis consists of two events, two important events. They are karyokinesis and cytokinesis. So mitosis cell division involves two important events. So the first event is karyokinesis, which is followed by the cytokinesis. So let us see what is karyokinesis. So karyokinesis is the division of nucleus into two daughter nuclei. Division of nuclei into two daughter nuclei is called karyokinesis. Are you clear? So, karyokinesis is nothing but the division of nucleus. When a nuclear division occurs, the process is called as karyokinesis. Are you clear? Now, it consists of 4 phases. The karyokinesis consists of 4 phases. Immediately, this nucleus cannot divide one into 2 parts. So, it moves on with 4 different stages or phases. We will see what are the 4 different phases. The first phase is prophase, the second is metaphase, third one anaphase, and the last one is the telophase. So, the division of nucleus occur in 4 stages. The division of nucleus occur in 4 stages or 4 phases. First phase prophase, next metaphase, then anaphase, and the last one is telophase. Are you clear, children? Now, let us move on to the prophase, the first stage of nuclear division. So, in the prophase, the chromosomes become short and thick. The chromosomes become short and thick. They are clearly visible inside the nucleus. So, the first stage here, the chromosomes become short, they become thick, and they are clearly visible inside the nucleus. Centrosome splits into centrioles. Centrosome split into centrioles and occupies the opposite pole. You know, always the centrioles play an important role in cell division. Yes, so here the centrosomes split into centriole and they occupy the opposite poles of the cell. Each centriole is surrounded by aster rays. They are surrounded by aster rays. You can see some lines over there, and the green color is the what? Centrioles. So, which is surrounded by the aster rays. Spindle fibers appear between the 2 centrioles. And here the nuclear membrane and nucleolus will get disappeared. Are you clear, children? I am repeating again. So, in the prophase stage, the chromosomes, they become short and thick. They are clearly visible inside the nucleus. Then chromosomes split into centrioles and occupy the opposite poles of the cell. Then each centriole is surrounded by aster rays. Then spindle fibers appear between the 2 centrioles, and finally the nuclear membrane and nucleolus get degenerated or disappeared. Are you clear, children, what happens in the prophase stage? Now, let us move on to the metaphase. In this stage, the duplicated chromosomes arrange on equatorial plane and forms the metaphase. You can see all the chromosomes are arranged in the middle of the cell in an equatorial plane. In a same equatorial plane, they are arranged in the middle of the cell. Each chromosome get attached to the spindle fibers. So, as I said, spindle fibers are already there at the pole regions. So, these chromosomes, they get attached to the spindle fibers by its centromere. The centromere of the chromosome gets attached to the spindle fibers. Are you clear? The chromosomes of the centromere of each chromosome divides into 2, each being associated with the chromatid. Are you clear, children? So, now let us move on to the anaphase. So, in this stage, the chromosomes What happens here? The centromere attaching the two chromatids. The centromere attaching the two chromatids divide and the two daughter chromatids of each chromosomes get separated. You can see the chromosomes which are arranged in the middle, they get attracted by the spindle fibers and they divide equally and they move towards the spindle fibers. They move towards the spindle fibers. The centromere of each chromosome divides into two, each being associated with the chromatid. So they get separated equally. Got it? Now, next is the telophase, the last phase. Each chromatid or the daughter chromosomes lengthens and become thinner and turns into a network of chromatin reticulum or chromatin threads. Now you can see the separation of the nucleus occurs. The chromosomes, they started behaving normally, that is they become thin and turns into a network of chromatin fibers or chromatin threads. Are you clear? The spindle fibers break down and they get disappears. The nuclear membrane and the nucleolus reappear again. It comes, it retains, it regenerates again in the each daughter nucleus. Are you clear, children? I think you are clear enough in all the phases of karyokinesis. Okay, now we will see cytokinesis. The cytokinesis is nothing but the division of cytoplasm. Once the nucleus gets separated, the cytoplasm should also get separated, no, should also get divided. So, the division of cytoplasm into two daughter cells by construction of the cell membrane is called cytokinesis. So, here a complete two daughter nucleus get divided into two separate cells. Are you clear? So, cytokinesis is nothing but division of cytoplasm. So, after the division of nucleus occurs, then it is followed by the cytoplasmic division. So, first nucleus should get divided, followed by the cytoplasmic division. So, cytokinesis is nothing but the division of cytoplasm after the division of the nucleus. Are you clear? Now, let us see the significance of mitosis. This equational division results in the production of diploid daughter cells. So, here the daughter cells will be 2n in number with equal distribution of genetic material. Both the daughter nucleus will have equal number of genetic materials, that is diploid cell, 2n diploid cell will be formed. Are you clear? In multicellular organisms, growth, organ development and increase in body size are accomplished through the process of mitosis. Are you clear? Mitosis helps in the repair of damaged and wounded tissues by renewal of the lost cell. So, when a cell get damaged, this by the process of mitosis they will again replace the older cells. Are you clear, children? So, this is the significance of mitosis. So, that is all. We come to the end of this session. We will meet you in the next session. Thank you, children.